Hi, good morning, good afternoon, good evening. I hope you're all well. My name is Ben Eggleton. I'm the director of the University of Sydney Nano Institute at the University of Sydney. My job is to introduce the session, but before I proceed, I would like to acknowledge the traditional ownership of the custodianship of country on which the University of Sydney campuses stand. I'm at the Camperdown campus, which is on the land of the Gadigal people of the Eora Nation. We pay our respects to those who have cared and continue to care for country. So it's great to be with you this morning, this afternoon, this evening for this fantastic session. Very briefly, the housekeeping appears on the slide. Some very obvious points to ask you to be muted. Questions will be addressed through the chat. It's recorded. We have a Twitter hashtag. We do ask you to reveal yourself through the camera. We find that when we can see you, we can engage with you, and we get some great screenshots for our social media. So please do let us know, put your camera on. So I'm going to say a few more words in a moment, but it's my pleasure now to introduce Professor Duncan Iverson, who is the Deputy Vice Chancellor of Research at the University of Sydney. Duncan is trained as a philosopher and is from the social sciences and humanities, but has always demonstrated a great commitment to innovation, technology, and sustainability, and been a great champion of multidisciplinarity. And Duncan's going to give a University of Sydney perspective to set the scene. Duncan, over to you.
Thanks, Ben, and good morning, good evening, good afternoon to everyone. Wonderful to be with you. And let me also acknowledge the traditional owners of the land this morning that I'm on. I'm also on the land of the Gadigal people, one of the first peoples in the Sydney Basin region. I'm just near the campus this morning. So let me acknowledge, it's a very important part of the legacy and the inheritance that the University of Sydney is very proud to share with the first peoples of Sydney. So let me acknowledge traditional owners this morning. And let me also just congratulate you on what looks like a fantastic program. And I guess your work over the past few days and today really touches on two things that are really important to the University of Sydney at the moment. The first, obviously, is the UN Sustainable Development Goals. And the university has really embraced those goals as an important aspect of the way we think about our research, our education, and indeed the way we work as a university. So we're very committed to the role that universities have to play in advancing the SDGs, and also thinking specifically about advancing the SDGs in the context of our part of the world in the Asia Pacific and the challenges that we face, but also playing our global role. The event today obviously is a great manifestation of Goal 17, Partnership for Goals. I'm sure you all, the numbers roll off your tongue. You're all thinking of the different goals that you're working with, but partnering for goals is a vitally important aspect of us actually making progress in this space, and universities have to work together and work with our industry and community partners in achieving that. The second thing is really, I guess, the university's commitment to nanoscale science and technology. And as Ben mentioned just briefly, and as many of you will know better than I, again, this is an extraordinarily exciting area of research and innovation. And the University of Sydney has obviously committed ourselves to a whole of university initiative in nanoscale science and technology. And Ben, of course, leads that initiative through Sydney Nano. And we're trying, like many of you, and like the University of Waterloo, a university which, we're a huge admirer of, we're really trying to harness our research and teaching in ways that create the conditions for greater impact for not only our students and staff, but for the communities in which we work. And there's no doubt that nanotechnology, nanoscale science is going to be a huge area of innovation and importance, and indeed in relation to advancing the Sustainable Development Goals, and also in relation to nanomedicine. Like many of the universities participating today, the University of Sydney has a large life sciences and clinical sciences footprint, and we see nanoscale science and technology as a crucial part of the future of medicine and health, and also part of the future of delivering better health outcomes for our communities. So I'm just really delighted to see the event today, delighted that we're supporting the Network for Sustainable Nanotechnology, and I'm really pleased that we're able to support your work this morning or this evening or wherever you are. So thanks again for the event today. And Ben, I'll hand back to you.
Thanks, Duncan. That's brilliant. I appreciate those comments and appreciate you being with us this morning. So a few more remarks from me to build on Duncan's introductory narrative. A little bit more about Sydney Nano. We were established in 2016, so we're a relatively new initiative at the University of Sydney, as Duncan mentioned, part of the university's commitment to multidisciplinary mission-directed research. And interestingly enough, our initial focus was indeed in quantum science and nanophotonics. And I had the pleasure yesterday to attend, Duncan would know, the Australian Quantum Conference that was downtown, which was attended in person by hundreds of people, which was fantastic. We're headquartered in the Sydney Nanoscience Hub, which is a state-of-the-art, world-class, world-leading nanoscience building with facilities, laboratories. But the point here is that about 4 years ago, we have put forward a completely fresh strategy that is indeed whole of university, multidisciplinary. And the UN sustainability goals have been front and center for all of our initiatives. As it relates to our Grand Challenge projects, our networks and other strategic partnerships with institutions like the University of Waterloo and Bina from Tel Aviv, and I can see our good friend Dror Fixler is joining us today. Some of our current priorities are indeed in the space of nanomedicine. Wouldn't surprise you to say that we're working on COVID-19 related research, particularly looking at aerosol transmission of COVID. We have strong initiatives in smart sustainable buildings and energy diversity. So this conversation today aligns beautifully with our Sydney Nano community. Finally, I want to acknowledge the very strong partnership with the University of Waterloo and the Waterloo Institute of Nanotechnology. Enjoyed working very closely with Shashank and Lisa and the team since we put together the network and of course this event. I also want to acknowledge the Sydney Nano team, in particular Rye Cleary, the senior research officer who has been with Sydney Nano for less than a year, done a lot of the heavy lifting to help with this meeting. And of course, my colleague Wojtek, who's here from the School of Pharmacy, who's been really a driving force for our strategies around nanohealth and nanomedicine. I wish for a great discussion today. Look forward to engaging you through the chat. Please leave your cameras on so we can see you and engage you. And with that, I'm really pleased to introduce Steffi Friedrich, the Director of Acumen IST, who will moderate the keynote speakers that we are hearing from this morning. Thank you very much, and over to you, Steffi.
Thank you, Ben, and many thanks for the introductory words and the kind introduction, and also to Duncan for setting the scene and the background of the university's implementation of the Sustainable Development Goals. Mirroring the excellence that has just been mentioned to you, we have brought together a panel of international speakers here. We have two keynote presentations from you. The first one from Terry O'Donnell from the US, and the second from Christine Allen. And with that, and not wishing to take any time away from the great technical discussion, I give over to Terry O'Donnell from Northwestern University.
Great, thank you, Steffi. Okay, so I just want to thank the organizers for allowing me to be part of this initiative. These are things that we have been thinking about for a while. I'm glad to have the opportunity to share a little bit how we're thinking about the sustainability goals. So I want to just provide an overview of what might be important in the design of constructs for nanomedicine. And in particular, I want to focus on the shape effects and how different aspects of ligand design can have an impact on developing the next generation of therapeutics. So part of the reason why nanomedicine is so attractive to the community is that it enables precision medicine with a very localized burst of agent. So for example, if there's a tumor in the lung, there's a way to access the tumor and then deliver it in a reliable fashion. But there are several challenges associated with nanomedicine and drug delivery. The first one is a delivery problem. How is the nanoconstruct and the nanomedicine going to be delivered? Is it systemically? Is it pill form? That's a macro scale problem. The second is once the nanomedicine makes its way into the body, can it target the region of interest? Or is it just going to be non-targeting like a lot of chemotherapeutics? The third aspect is a second delivery problem. Once the nanomedicine is able to actually meet the target, can it deliver? Can it deliver its payload? Does it still work? And especially this idea of the mechanism of action, maybe the particle gets there, but maybe in its transit there, it's accumulated a lot of proteins, a non-specific absorption of biomolecules that might impede its mechanism of action. That's another important question. For inorganic particles, the idea or discussion of whether a particle could clear or will it have deleterious effects if it remains in the body is also a question of interest. So today I'm going to focus on the fundamentals of the second delivery problem. So we are interested in these anisotropic nanoparticle shapes because we believe that the local nanoscale structure can produce very interesting macroscale chemical and physical properties. And this all relates to curvature. So the different local curvature regions may affect ligand loading and the overall construct properties. And I'll highlight these specific examples as we go along. First, these regions of positive curvature, they can exhibit high local optical fields when light is illuminated on them, but they also splay out any type of ligands that can be on the surface. It can retain the ligand functionality. Second, there are these regions of neutral curvature. So depending on how the ligands are covalently bound, you can have high packing, especially of thiolated ligands. And in terms of these negative curvature regions, we have these pockets that, because of its steric effects, can create local hydrophilic or hydrophobic regions, depending upon the types of ligands that we covalently attach to the surface. So there's a lot of the phase space is pretty large to be able to tune these designer nanoconstructs. And moreover, because they're made out of gold, they scatter light very well, and they can be used as optical probes with multivalent interactions. Multivalency based on the ligands that are displayed at these positive curvature regions, but also these anisotropic structures that can potentially bind multiple tips at the same time. And we can track what these multiple tips are doing in optical microscopy. So the first important point, at least related to some sustainability aspects, is the synthesis of the construct needs to be biocompatible. And so we start with a goods buffer, the goods there, which is heaps. There's a set of 20 goods buffers that can maintain pH levels and physiological conditions. And we use this as a reducing agent of this gold salt precursor, as well as a directing agent. So we have one molecule that does two functions. And this will be a theme as we go throughout the talk today, is how can we simplify the design of constructs so that they can be scalable and there's less waste and more uniformity of the product at the end of the day. So we can scale up the synthesis of these particles. This is an example of 5 liters of gold nanostars. These are low resolution transmission electron microscope images. And you can see that they're not all identical, but that's expected given that it's a seedless reaction and the reaction is done at room temperature. But more importantly, if you look at the scale bar and evaluate the length scales involved, you'll see that the tips of the stars taper down to the size of a protein molecule, 3 to 5 nanometers. So in principle, individual tips can interact with individual molecules. But the most important work after we can synthesize these particles in high yield is to be able to functionalize them with ligands that can do more than one thing. So we want to, or our focus has been to design ligands that can not only target the receptor of interest in a cell, but also perform an action, can function as a therapeutic. So we've focused on different types of thiolated molecules, such as single-stranded DNA, double-stranded sRNA, and these different types of aptamers, which can be thought of as chemical antibodies, so they can fold in a way and bind to a protein target. So we're interested in molecules doing multiple functions. But then the other important point that receives very little attention, but needs more attention, is how can you quantify and tune what's on the surface of the particle? And so we have found a way to tune the number of DNA strands on a single particle using a citrate buffer at low pH. And the reason that you need to use a citrate buffer is that DNA has these negatively charged backbones. You have to find a way to load more and more ligands onto the surface. With a citrate buffer method, we can take a 2-day salt loading process and reduce it to 10 minutes. That's a big savings in human effort. And also the excess DNA that we need can be reduced by 1/3. But the other big portion that we need to think about is we need to ensure that the secondary structure of the oligonucleotide in solution is maintained on the particle surface. And so this is actually a lot of work to ensure that the regions that are folded in terms of G quadruplexes in these cases, or in the case of the duplex, are maintained. So the slide summarizes like years of work that somebody has to do in order for us to have confidence in the therapeutic effects. And so we did it. So these nanoprobes, as we will characterize, can also function as drug carriers. And so for this particular construct design, I just want to emphasize some of the advantages of using gold. Gold nanoparticles themselves are non-cytotoxic as long as you don't start with cytotoxic ligands. And so this is why we started with the HEAPS buffer. If they're functionalized with DNA or RNA, it improves the stability against nucleases when you introduce these to cells. You're able to achieve a higher cellular uptake through endocytosis, through multivalent effects, and also having all of these ligands on the surface, the particles preserve and enhance the DNA function. But in terms of the structural effects of the nano constructs, it's less clear on how these local changes in either ligand density or size and shape will affect the therapeutic or intermediate outcomes. For example, the size and shape of the nanoparticle core, the ligand type, whether it's targeting or not, the ligand composition, as well as the density. So today we'll focus on 2 aspects. The first is the size and shape of the nanoparticle core, and the second is the liganding type, whether it's targeting or non-targeting. So an important question that we need to evaluate is whether the targeting nanoconstructs retain their properties in vitro, because we can spend a lot of time optimizing these nanoparticles on the benchtop, but once they go into cells or once they go into animals, they might not work. So we're interested in a couple of different types of nanoconstruct protein interactions. The first is to, again, use this drug molecule that attach to the particle that can also bind to receptors on the cell surface. So the idea is we have this construct, we put the ligand, and then we have this nanoconstruct that can bind to receptors on the cell membrane, such as HER2, or even to membrane receptors that are not transmembrane in terms of nucleolin. The other type of nanoconstruct protein interaction we would like to do for targeting ligands is we would like to have an adjuvant related to stimulation of the immune system, and whether this adjuvant could bind to a protein target within the endosome. So we're looking at 2 different classes of interactions on the surface of the cell membrane and interior inside an organelle. But the big problem is potentially this. What about this concept or this idea of what's called a protein corona, and will it impede biological effects? I alluded to this earlier. We can optimize everything on the benchtop, but then once we put them into serum or blood or any other type of way to deliver the constructs in vitro, you're going to have non-specific absorption. And so the literature is mixed on this problem and how you can take advantage of it or overcome it. So the early work stated that the protein corona fingerprinting predicts the cellular interactions. So if that is the case and that's driving it and you don't care what the ligands are, then all of your work is, maybe less useful. There's been studies related to the molecular structure and some studies at the single particle level. Does the molecular structure of the protein corona affect cellular outcomes? And this recent review article that was just published in ACS Nano, scratching the surface of the protein corona, challenging measurements and controversies. So this is an important fundamental problem that nanoscience still needs to solve, but it has huge implications if we're gonna, if these constructs are actually gonna be useful. Okay, so there's some unknowns. The protein corona seems to control or seems to have some influence on interactions of cells with nanoparticles, but the literature is fairly limited about protein corona effects on targeting interactions. And I introduced 3 different types of targeting interactions that we wanted to focus on because we wanted to investigate real systems. Okay, so the first one is we're interested in targeting the transmembrane receptor HER2, which is overexpressed in breast cancer and other types of cells. We're using this aptamer with this particular sequence. You can see it's a trimeric version of a 14 base pairs. The 14 base pairs was not invented by us, but it was published in other work for human studies. And basically, if you just have the 14 base pair, it will bind, but there will be no therapeutic effect. So we needed to take advantage of the trimeric version of this. So there's some multivalency involved. We wanted to compare at the therapeutic level, which is about micromole for aptamers, with what is possible on the nanoconstructs. So we first compared the uptake for the free aptamer versus the aptamer on the gold star. And you can just see by fluorescence that there's a lot more of the fluorescence because more of the particles are taken in and the aptamer is not degraded. If we look at the cell viability, which is just giving you an indication on metabolism, you can see that there is very little influence on the cell viability of the free aptamer, but once it's attached to the gold nanostar, there's a significant reduction and an increase in cell death. But the mechanism of action of this H aptamer is that it cross-links and sorts the HER2 and sorts it on the cell surface and transits it to the lysosome for degradation. So this is the mechanism of action in which we would like to validate, at least for the gold stars. So you can see that if we stain for HER2 after treating these cells with free aptamer, there's still a lot of HER2 left on the cell membrane. But once we treat it with H aptamer on the gold nanostar, there's a significant reduction in the HER2 just based on fluorescence. Of course, these are qualitative measures, and so we needed to evaluate this quantitatively using Western blot analysis. And if you compare it to the housekeeping protein beta actin, you can see that there is a reduction of the HER2 overall based on being exposed to the nanoconstruct. So this means that our system works, at least in these cells. Okay. So how can we compare this to some imaging analysis? So we're using optical microscopy, and in this case, differential interference contrast microscopy. It's a phase contrast imaging, so any big changes in refractive index, that's what you will see. And these are just different slices of what the cells look like. These are live cell images. So how do we differentiate the gold nanostar construct in the mix of all of these complicated cellular structures? Well, the good news is that these gold nanostars, they are anisotropic in shape, meaning they're very sensitive to polarization. So for example, if this long axis of the nanostar is oriented along 0 degrees, the particle will appear bright in the far field. Similarly, if the particle has rotated 90 degrees, it will be dark in the far field. So we can correlate these nanoscale structures with the optical images. And so what we see is that if we compare this targeting construct with a non-targeting construct, there are 2 different pieces of information we can uncover. One is just looking at the contrast and what I told you about in terms of rotation. And then secondly, you can look at rotation as well as translation, all in the same system. So this is the targeting HER2 construct, and this is the non-targeting anti-HER2. It's just the scrambled sequence. And you can see just based on translation that the H aptamer construct that's targeting spans a very large range compared to the non-targeting, which is mostly confined. And so what does that mean also in terms of rotations? Well, what's quite interesting in terms of the rotational motion is that there's a lot of rotational frequencies that these targeting nanoparticles will undergo compared to the non-targeting. So the targeting nanoparticles show exceptionally different dynamics, translational and rotational dynamics compared to the non-targeting, which is important and which is distinct because the protein corona is the same for both of them. Okay, the protein corona is the same. We did that analysis, but now in this case, they still work. The targeting particles look really different. I won't go through all the analysis of the translational dynamics, but just to show you the statistics in comparison. So we found that restricted diffusion is most common, and this gives you ideas on the confinement length. And if you compare the confinement lengths for targeting and non-targeting constructs, you'll see that we extract a confinement length L, which is about the size of the HER2 domains. And so the HER2 basically needs to transit within this length of time in order to bind and be taken up intracellularly. So this is what endocytosis looks like of a single nanoprobe. This is what the particle is on outside, and it's rotating, and it stops rotating because it's bright, and then it just transits inside. So I'll just do it one more time. So it's rotating as it's finding the receptor, and then after a while it stops rotating. It just stays bright for a couple of seconds, and then it translates inside cells. So in terms of the mechanism, there's very little translational motion for 20 minutes, at least once we saw this particle. And then the brightness does not change for about 12 seconds. And then after it's internalized, which takes less than a minute, then there's rapid directional motion, probably along actin microtubules. So we start to identify these different length scales and these time scales just based on these single systems. Okay, in the last couple of minutes that I have, I just want to show some brief examples of shape effects because I think these are quite interesting. So the first is this is the second membrane receptor that I mentioned. So these particles combine to nucleolin, and the nucleolin can shuttle the nanoconstructs to the nucleus. Turns out the nanoconstructs by themselves produce late-stage apoptosis, but because these particles are made out of gold, you can shine femtosecond light on them, and you can release the aptamer that can then thread its way through the nuclear pore complex and result in significant cell death. This is DNA damage, and this is the change in phenotype as with all the released drug. And so we want to determine the shape effects of these targeting nanoconstructs. So we're going to be using nucleolin positive cells, and also we can block nucleolin for nucleolin negative cells. So we needed to make sure that the physical and chemical characteristics were the same. So we did this as much as possible in terms of surface area, DNA density, zeta potential, dynamic light scattering sizes. And we also, again, measured the protein corona on these constructs, and you can see that they're very, very similar. So any changes that we do see in the dynamics must be because of particle geometry. Thank you. And so the first thing we needed to establish was the ability of these gold nanostars to bind nucleolin. And these are what the optical images look like. And what you can see if we evaluate the contrast is that it fluctuates a lot if the nucleolin is blocked, but once it's not blocked, then they can interact. And these are the different diffusion modes that you can see here. They're very different from what we observed earlier. In this case, we evaluated a lot of particles, just all these single particle data analysis. And the distribution of these different modes is quite different. And I won't show you the statistical data, but these gold nanostars show almost twice as fast a velocity, as well as twice as large confinement. So these are very distinct features, which also it indicates that these different constructs bind to different receptors, with the gold nanostars binding to nucleolin. Okay, so I'll skip these other effects in the endosome just because I'm running out of time. I'll just do this here. Okay, so I introduced the anisotropy as a function for designing these interesting constructs. This is a green synthesis, which is biocompatible. These excess DNA ligands that we need for functionalization can be reduced, and now DNA ligands are easily synthesized. These ligands are designed to be at least dual functional, and we can also use light as a non-cytotoxic stimuli. In terms of these nanobio interactions that we then can correlate to a therapeutic response, we find that the targeting and non-targeting anisotropic particles show very different dynamics. This is a new type of dynamic assay. And also the targeting nanoconstructs of different shapes show different dynamics, and they also bind to different proteins on the cell surface. So the key takeaway point is that the shape, the anisotropy in shape, is very important to retain the targeting effects, because the protein corona is still there. We've already validated that, but still the anisotropic particles are able to function while the spherical particles are less so. And this is the group. Many of them, a couple of them contributed to the work, but I'll stop there since I'm out of time. Thank you.
Thank you, Terry. What a tour de force that was. And I'm sorry you had very little time to show all of your fabulous examples and results. I do, however, want to take the time to give the audience one question, really burning one. If not, I think I'm gonna have to ask you one, and I'm thereby I may be taking away a little bit from the panel discussion, but we can continue that there. So what we have on this side of the Great Ponds is the concept of safe by design, which has now morphed into safe and sustainable by design. It is a thing. It has capital letters in it, and it is it's about to be written somewhere into a European policy. So I wonder what would be the first knob that you would turn in order to make something safe and sustainable by design? I mean, you have a great number of parameters to choose from. What would be the first one you would look at?
Yeah, so some of ours, we had that in mind. So we wanted to focus on fundamentals, but we also knew it needed to be scaled, and then it needed to be able to undergo like FDA approval if the construct was ever going to work. And so that's why we focused on, say, ligands that could do more than one thing. And now that there are DNA synthesizers everywhere, they're more ubiquitous. That's not a problem. But also the method of synthesis. So there's a trade-off between, if you want all your particles to look identical, that means you typically need to use something that's seed mediated, which adds an extra step. And it usually also includes cytotoxic ligands. So for example, this is why we don't use nanorods, because it's impossible to get all the C-tab off. And we really just want the simplest design, the simplest multifunctional construct that we can have, which is why we actually focused on this particular design and this particular size, because it's not that large. These are like 30 to 50 nanometers tip to tip. So there's different length scales that are going to be important related to whether you have unwanted accumulation in organs, for example.
Thank you. And I think that was a perfect introduction to Christine's presentation just now. Christine Allen, the Associate Vice President and Vice Provost at the University of Toronto, will pick up on what you just mentioned, in that you say that the method of synthesis is a very important one. And I think what could be more appropriate than hearing what automation can bring to exactly that part for sustainable drug design. Christine.
Fantastic. I will just share my screen and hopefully I will be successful in sharing it. There we go. Is that okay? Can you see my screen?
Yes, we can.
Yes, we can.
Perfect. Okay, just wanted to make sure. Thank you. So delighted to be here today and to have the opportunity to connect with you online. I'm sharing with you some of the work that we've pursued in collaboration with Professor Elias Berguzik, he's at the University of Toronto. And this is work that we began really at the outset of the COVID-19 pandemic, and in part was motivated by the challenges that we were facing at that time. So when I think back to early 2020, before the COVID-19 pandemic really hit us in Toronto, and then 6 months later, even now, and what's changed in our lives, one of the most striking differences for me is our embrace of technology in our daily lives. Technology, technological advances, just permeate our lives now more than ever. As an example, I'm a bit of an exercise fanatic and regularly went to the gym or fitness classes, and am now able to do near to all of this through online platforms. We pivoted all of our classes online at the University of Toronto for over 100,000 students. These were pivoted online in a matter of days. And as you know, we've been holding meetings like this one and seminars online. We're doing our shopping online. Doctors' appointments are now more readily available online. So technology has really been a key enabler during this time. At the same time, during the pandemic, in addition to technology, we've seen the impact of drug formulation and delivery, with 2 of the first vaccines to receive clinical approval in North America relying on delivery in lipid nanoparticles. And these were approved in less than a year from the first report of the SARS-CoV-2 virus, making what would have seemed impossible at the time possible. And frankly, I'm not sure there's been a more exciting time to be a drug delivery or formulation scientist. My joke usually is, for the first time ever, my parents understand what I'm doing now. They understand what a nanoparticle is, they're interested, they think it's important. So our current formulation development approach, however, often continues to include far too much trial and error. To take one example, this slide shows a hypothetical development pathway for a polymer particle-based formulation of a drug. Typically, we begin by identifying criteria that the formulation must meet for use in a specific application. We then make several formulations, characterize them, and evaluate which meet the designed criteria. Of course, we often start by reviewing the literature or a formulation work that we've conducted in the past, yet still the development is time-consuming and resource-intensive. So in this day and age, there must be a better way to do this. We started to think about integration of AI and machine learning into our drug formulation efforts, and the way in which they may provide a solution to fast tracking drug product development and to foster innovation. So in my case, when the pandemic hit, the lab was completely shut down for 2 months and then open with physical distancing restrictions in place. The restrictions then loosened and they tightened and they loosened again, and we'll see what happens in the future. This motivated us to use this time to embrace a number of new approaches as a means to really accelerate and drive our drug delivery efforts forward. So as part of this, we began to integrate AI and machine learning approaches, and we also began to adopt automation as a means to accelerate the acquisition of high quality data. So what is artificial intelligence? How will artificial intelligence impact us, or how can it impact us as pharmaceutical scientists? Will it and can it replace us? There's lots of hype, excitement, in some cases confusion and fear. And a book that I really love that I think puts AI and specifically machine learning in simple terms is called Prediction Machines. It's written by 3 professors at U of T, AJ Agrawal, Avi Goldfarb, and Josh Gans. And if you don't know what if you don't want to read the book, you might enjoy watching some of their talks online on YouTube. But in this book, they explain that this latest wave of AI does not actually bring us intelligence, but instead a critical component of intelligence, prediction. Essentially, that at this time, machine learning, which is a branch of AI, is considered a technology of low-cost prediction and discovery. So if we think of machine learning as a door to low-cost prediction, as a drug delivery scientist, we can start to think about the things that we'd like to predict. So for me, it's which excipient or material should I use to formulate a specific drug? What will the drug loading capacity be for a specific formulation? And even better, what will the release kinetics of a particular drug be from a formulation? There's so much that we can predict. So AI and machine learning are some of the hottest buzzwords of our time and are said to have the potential to revolutionize many industrial sectors. And in recent years, we've certainly seen the pharmaceutical industry utilize these tools, especially for drug discovery applications. So what about AI and machine learning for drug formulation development? A little over 2 years ago, back in October 2019, we wrote a perspective on this topic, and in it we highlighted the increasing number of publications that really encompass some form of computational prediction or a simulation element. And we've since updated this now, and you can see that there's really only been an increase in the number of publications in this space. So I'm going to give you an example of how we've begun to integrate this into our work. So we've been working in collaboration, as I said before, with Professor Asper Guzik to explore the use of supervised machine learning to predict the properties and performance of drug formulations. So how do we approach this? First, we begin with data set construction. And for this, we either use experimental data that we've generated in-house or by extracting data from published studies. And usually we use a combination of both. Once we have the data, we need to numerically describe each experiment using input features. So these can be the physicochemical properties of the drug. So things like the log P of the drug, the molecular weight, the water solubility. And we do the same for the material or the excipient, as well as the processing parameters that are used to prepare the formulation. And once we have a well-described data set, we divide it into a training set and a testing set. We can now begin the process of selecting a modeling strategy. And a large number of these machine learning models are actually available in established open access libraries. So typically we will train a few different model architectures, things like random forest, linear regression, neural network models, and we then evaluate these models to find the best machine learning model for our data set. To evaluate the models, we compare the predicted test values with the actual or experimental values using a metric such as the coefficient of determination. And once we have a model that can be used to predict the desired properties with the appropriate degree of accuracy, we can assume that it has found some relevant statistical correlation in the data. And the model can now be analyzed to gain insights into which features the model considers most important for prediction of specific results. This is called feature importance analysis. So we currently have a few different projects that are going on in this area, and I'm going to describe one of these to you today. And actually, this one is focused on the development of polymer-based, long-acting injectable formulations, such as microparticles. We also have a number of nanoparticle projects, but I'm sharing findings from the microparticle project with you because it's further along. So the motivation for this project came out of two recently published papers that describe studies conducted in collaboration with an industry partner and were focused on the development of long-acting injectables that were formed from new polymer materials. These papers represent years of work, and some of the most time-consuming aspects of the work, as you would know as formulation scientists, are really execution of the drug release assays, which in our case took up to 3 months at a time. During this project, we would prepare and characterize the drug-loaded microparticles, and in some cases only, we're disappointed with the in vitro release behavior. And then during group meetings, we would often discuss how in this day and age, there must be a more efficient way to develop these types of formulations. So with this new approach that relies on AI and machine learning, I actually believe we could have cut down on the time, the money, and the resources that were necessary to conduct the research described in these papers. So for the research I'm sharing with you today, we extracted data from 40 published papers. Briefly, this data set was constructed using, as I said, 40 papers that provide about 200 in vitro release profiles. And about a third of these are release profiles from our lab, or were measured in our lab. So they're our papers. This data was then divided into a training set, and the training set included about 100 drug release profiles, and a validation or a test set that included about 80 drug release profiles. Using the training data set, we then trained a series of machine learning models, again, taking each of the experiments or formulations and describing it in terms of numerical terms or input features. We trained the models. And then we assessed the limitations of the trained models with a second data set, which I refer to here as the validation data set. And with this second external validation data set, we could apply our newly trained model, assess the performance, and try to identify limitations. In the validation data set, 50% of the drugs and 80% of the drug polymer combinations were unseen by the model. So we screened a series of different machine learning models to select which best fits the data. So here I'm showing you the machine learning model performance in terms of mean absolute error. Each of the models is represented by a different color. We've got random forest model, decision tree model, a neural network model, and so on. If we look at the data for each model, each of these white dots here represents the absolute error in predicted relative to actual drug release for a single experiment. So there's about more than 1,000 dots represented here in each model. And we can see from this that the random forest is the model that gives us the best prediction accuracy for our data set. The average prediction accuracy for the RF model, the random forest model, was 0.17. So this means that on average, the fractional drug release predictions were within 0.17 of the experimental values. The good performance of the RF model is interesting for us, given that it's an older and a relatively simple model. By contrast, the neural network model is a more powerful and advanced model, but it also is more complicated and likely requires more data than we currently have in our data set. So here I'm showing you the feature importance analysis for the random forest model. The model is able to identify which features have the greatest influence on the output we're trying to predict, in this case, fractional drug release. As you can see for this random forest model and this data set, the polymer molecular weight has the greatest influence on the drug release predictions. And then the other input features here are ranked in descending order of influence. This is a summary of how the model performs on our validation or test set for the different drug polymer combinations. The average overall prediction accuracy of their RF model, again, was 0.17. And you can see here that in general, the individual drug polymer combinations are below or similar to the average. However, we can see that a few of the polymer drug combinations, for a few of these drug polymer combinations, the model is less accurate than the average. So before I get into that, because that likely reflects some limitations of the model, I'm going to go through a few of the examples of the predicted drug release profiles relative to the actual or experimental drug release profiles. So I'll show you the profiles for the different drug polymer combinations that I'm showing you here. So here you can see we have a few of the drug release profiles with the predicted profiles in blue, experimental in yellow, with the blue halo representative of the confidence interval associated with the predictions. To generate the confidence values, we've used a previously published method that is shown here. And so you can start to imagine the potential applications of this model. For example, we can now conduct virtual screening of different combinations. Drug you want to formulate, you've got a series of polymers, you can plug them into the model before you begin experimental work. And this will allow you then to shortlist promising drug polymer combinations. And from that perspective then to reduce the overall trial and error approach to formulation development. In general, I believe that this integration of such machine learning models has a role to play in making our drug formulation development process more sustainable. So if we kind of look at this release profile here, where we see relatively poor, quite poor prediction of our experimental values, and we want to dig a little deeper to understand why that's the case, we now go and look at the properties of this drug relative to the properties of the drugs that were used to train the model. So for this, where we saw this relatively poor prediction, this was gefitinib in a PLGA microparticle. So this slide shows a breakdown of the select physicochemical properties of drugs that we use to train the machine learning models. The white dashed lines are this normalized distribution overlay. And using this approach, we can see where the physicochemical properties of this outlier, or gefitinib, in this case, lies within the histogram of the training set features. So as you can see, for the most part, gefitinib falls within the histograms, with the exception of its pKa. Right, so here we see the pKa of gefitinib is quite low relative to the drugs that were used to train the model. But beyond this, this type of analysis can actually be quite useful in terms of identifying ways to improve the model. We can see where we have gaps here, and we could then select drugs that will help us to fill these gaps to strengthen the model for future predictions.
2 minutes.
Thank you. With that in mind, in terms of our training data set, one of the challenges that we've run into and that I hadn't fully expected has been collection of large volumes of high quality data on polymer microparticles. And frankly, we've seen the same for nanoparticles, and this has really delayed that project. It may seem like an easy task to compile a large volume of data on polymer microparticle formulations of drug, given that they've been studied for decades by our community. However, it's not as easy as you think. So in this table, we've summarized data from 10 formulations that were included in the top 20 cited PLGA microparticle formulations over the past 20 years. So you can see here specifically specific pieces of information that are missing. And as I mentioned to you previously, we require these specific input features or characteristics of the formulation, in order to train the models. So this is quite frustrating. In order to create the training and validation data sets, in fact, we combed through more than 200 papers to find 40 with complete data sets that we could use. So this highlights one of the most significant challenges that we have in science now. As I said before, when we first began this project, I chose microparticles in part because we've been working on them, but also because I knew there was this vast, array of literature out there that we could draw from. And obviously it would be a real shame not to be able to build on the work that's been done by others. However, as you see in that last slide, our ability to do this is limited, to some extent at least, reporting or heterogeneity in the reporting of scientific data. Critical properties of a polymer. Okay, can you hear me? I'm not sure. Can you hear me?
It is breaking, but now
It's
I think it
breaking?
is okay. Now it is okay because
Oh,
the
sorry. I'm so sorry. It's okay now. So
Yes.
I'll just quickly sum up because I know my time is running out. But just to say that I think, thinking about this and the reproducibility crisis, well, I think we start to think about, why we're publishing our data. And it shouldn't just be about increasing the number of publications or citations. It should be to share our findings, to advance the field, to enable others to build on the work that we've done. So in recent years, there's been a significant emphasis on sustainability, and sustainability can be defined as meeting the needs of the current generation without compromising the ability of future generations to meet their needs. And it seems highly wasteful and irresponsible not to be generating data and reporting it in such a way that it's useful to future generations. So I won't expand on the automation, the integration of automation that we've been doing in our work. And this is really to help us, collect large volumes of high quality data. And to some extent, it's also helped us to accommodate COVID-19 restrictions. This is just a timeline of some of the work that we've done through the years. This is something I've been, really keenly interested in removing the trial and error approach to drug formulation for decades, frankly. And we've, pursued a number of different approaches to do this. I would say that from our experience so far, machine learning is widely accessible. It's relatively inexpensive, both computationally and economically. And many platforms, models, and algorithms are open source. And we're also committed to sharing all of our outputs of the research, the trained models, the databases, codes online for other research groups to use. So a quick shout out to my collaborator, Alain Esper-Guzik, and our group for the work that we've done in this area to this point. And thank you for your attention.
Thank you, Christine. Thank you. That was, again, quite a powerful tour through all of your tools and different validation methods. And I'm sorry we couldn't see more about the automation at the end, but maybe we have a chance to dip into that during the panel discussions. I think the organizers would like me to actually move all of the questions to the panel discussions. So we are taking a mini break now, and we are back here at the original time that we had in the program, which would be exactly at the hour, right?
I think we can take 10 minutes break, 10-minute break. That's absolutely fine. We can meet at 5 minutes past an hour, which will be 5 minutes past 8 in Sydney time. So 10-minute break.
to the panel? Ten minutes at our home desks seem to pass even faster than if we were at a conference trying to get a coffee in a very short break. But I think we have most people back again. So if I could ask our panelists to switch on their cameras again and prepare themselves for another interactive 55 minutes. We have 2 additional panelists for you that are definitely rounding the picture here of going around the world today in our nanomedicine session. One of our additional panelists is Samuel Shigome from the Nanomaterials at Botswana Institute for Technology, Research and Innovation. And our second additional panelist is Dror Fixler, the director of the Bar Ilan Institute of Nanotechnology in Israel. What I would like to do to brighten the eyes a little bit so that people have actually heard you and know what you think and how you think about this is I would like to give every one of you, of both of you, just a moment to introduce a statement about your view of this so that it is easier to then ask a question directly to you once we give over to the panel. So maybe in that order in which I just read it out, I'm not quite sure, do we have, there he is. Samuel, would you like to give us a short statement of reaction?
Okay, thank you very much, Steffi, for that. As mentioned, I'm in Botswana in the Nanomaterials Division. We, in terms of the kind of work that we do, we mostly focus on nanofiber-based materials. And in terms of nanomedicine kind of work that we do, is mostly focused on the diagnostics aspect, where we look at how we can be able to come up with point of care devices for early detection of from both the animal and human health. That's the most of the work that we do. So I think in terms of the 2 talks by Terry and Christine, I think from my perspective, it's something that is very important. What Terry talked about, especially if you talk about the green approach of synthesis of nanoparticles, and I think it came out very clearly that there is the aspect of taking advantage of the polarization ability to be able to easily pick up the nanoparticles that she's talking about. And also I think the aspect of biocompatibility, because I think if you look at, I think from the African point of view, there's quite a bit of work that people are doing where they're looking at the green synthesis approach of nanoparticles. So I think that is got a component of sustainability. It also is a component of biocompatibility as well, because I think it came out very clearly that the choice of precursor material of your nanoparticle and what you use as a capping agent and reducing agent is also important. So for me, I found that to be extremely useful. And I think the talk by Kristen, definitely a way of simplifying the whole process to move away from a lot of trial and error. I think that really improves on the efficiency in which we do things. Of course, you may say that in a way, COVID-19 was a bit of a challenge for the whole world, but I think you brought it out very clearly that you were able to extract the positive things out of it. So for me, of course, you were talking about the formulation, but as I was listening, I could see a lot of opportunities for a broad area of application of the same concept of being able to take control of simplifying the whole process of identifying the best combination that you can be able to come up with. So those are my initial comments. Thank you.
Thank you. I was wondering if we should give the summary of the entire panel presentation to you instead. That was an excellent summary of what was discussed. Thank you, Samuel. I want to give Dror the possibility to also come up with a statement.
Thank you very much, and thank you for inviting me here. It's almost midnight in the Middle East, in Israel. As we are in BINA, Bar-Ilan Institute of Nanotechnology and Advanced Material, we are covering all aspects of nanotechnology, the biology, nanomedicine, and precisely the bioconvergence, as we are calling it. It's one of the hotspots in these days. We have several directions to support this kind of thing. First is research. As we are a university, we are supporting our researcher, the 68 PIs, to do their research, and large part of them are coming from life science, bioengineering, chemistry that dealing with drug delivery. And as we analyzed during the last 12 years, when we are taking different disciplines and working together, it's not only collaboration, it's generate new science. A good example for that, it's optogenetics, some combination of optics, biology, neuroscience, and new segment of science came up in optogenetics. It's more than 10 years old, but in these days we are combining smart materials, engineering, and computer science in order to build the next generation of science. So this is from the pure science. As we are working with more than 150 companies, large part of them are pharma as well as medical devices. And when we are supporting them, we're purchasing equipment like advanced cryofib, EM, electron microscopes, developing processes for better analysis of materials from life science, and working tightly coupled with the Faculty of Law and other departments in the more human science to make it really deliverable to the real clients, to the citizen all over the world. One nice example of combining the research and the collaboration, it's with our great host here today, the University of Sydney. We announced about collaboration between BINA and the University of Sydney, Sydney Nano, and one of the projects that we are sponsoring goes to the direction of bioconvergence targeted drug delivery platform for ovarian therapeutics. And this is one PIs from each side, and we are continuing this direction. So we are supporting science, we are supporting industry, and we are supporting the combination of both of them together.
Thank you. That was an excellent statement too. I think what we're going to have is a very interesting round of panel discussions now. We have a couple of questions that people from the audience asked earlier regarding the technical talks. And I wonder if we can stay on topic a little bit and take the one that came in last first. And that would be a question specifically if with all of this great development that we are putting into and the great thinking and trialing that we're putting into the different nanoparticles and their loading in nanomedicine, we seem to fail at the clinical stage. And the question that one of the audience members asked was if what Christine presented, and I think that also goes a little bit into the direction of what Dror just hinted on, if all of these fabulous new technologies weren't giving us a little bit more of a handle to be foreseeing the reactions in clinical trials so that we can minimize the failure rate at that end. I wonder, Christine, do you have a comment on that one?
So just in terms of the work that I've seen and the work that we're doing, We're already using data for pharmacokinetics in vivo. So looking to see if we can reliably predict the pharmacokinetics of nanoparticle formulations or drugs that are administered in nanoparticle formulations in vivo, which is obviously then a big predictor of things like toxicity and efficacy. But I'm also someone that has been watching the many failures that we've seen where nanoparticle formulations look fantastic in vitro and even fantastic in many animal models. And then they go into phase 1 clinical trials and fail. But if it's in the cancer space, which many of them are, I think one of the challenges is our models don't recapitulate the advanced and complex stage of disease that these formulations are often encountering in phase 1. Right, phase 1, it's not these are not early stage cancers. In these phase 1 trials, it's usually cancer patients that have had however many other types of treatment, and this is the last best hope or potentially a last best hope. So they have advanced disease, complex disease, resistant disease. And that's often not, those are not the models that we're using in our animal studies. We're using, tumor models that are created from a single cell type that are in immune compromised animals that are, not metastatic. I think one of our greatest issues right now in this field is we're using very simple models. We get great data, and then they just don't translate, obviously don't translate and probably shouldn't be expected to in vivo. So that's where I think there's a big disconnect there. But coming back to the machine learning piece, our starting point right now is just prediction of pharmacokinetics.
Super, thank you. I wonder, or you have, yeah, there he is again. I think what you were hinting on goes into a similar direction as well. Do you have a comment, please?
Definitely, yes, I'm fully supporting that. And the claim that it's not working yet in practical, no doubt that we are struggling that. I assume you know Nomi Hallas, that was one of the initiators of this direction. She faced similar problems, but she already got some approvals, confirmations, and it looks like that there is a new direction to use it. But we need to remember that this is good for us. The reason that it's not working yet, it's that we need to make sure that it's good for us, for the human being. And at the end, we will find the correct direction. I mentioned before that we established a new department in our Nano Center for Smart Materials as a requirement to synthesize this new element for the new functionality. It's not only gold, no doubt about silver, but it's not simple gold. And in these new materials, I believe that based on the needs and the trial and error, we will find the direction to open the challenge. And we are in this direction, and we are doing a lot of efforts, investing money and time, and we will have it, I believe so.
Thank you very much. Terry and Samuel, I wonder if you have comments on that question, what we should be using in terms of technology in order to improve the success rate of going from laboratory models to in vivo and clinical trials.
Yeah, I can just say a little bit about why we've gone backwards, meaning we did scale up for animal studies. And we did them, and they're expensive. And the problem is, well, the good news is there's no toxicity, but the bad news is that with only small numbers of 6 to 12 animals, for a pilot study, you can't really draw a lot of conclusions. I mean, you can get a result, but then you can find any way to explain it. And I think that was a problem for us. I mean, when we did these biodistribution studies on rats, and rats are big too, they're huge compared to immunocompromised mice. There was a big difference in circulation, for example, between male and female rats. So already the sex of the animals makes some difference, and and so the challenge is then once we implanted tumors in the mice, the particles would make its way there, but then there's some sort of we don't know what's happening at the second delivery problem. So the first one, if it's systemically delivered by tail vein injection, which is mostly how we've done it, there's accumulation all across organs by something called the enhanced permeability and retention effect. So you don't even care about targeting. Particles just get there. But then what is the requirement for them to produce a very large therapeutic effect? And what level of that is acceptable? And I just felt like after, tens of thousands of dollars of the animal studies, I mean, we'd have to be all in, meaning this is the only thing your lab did. And we work on a lot of different things. And so I felt like that sounds like, well, I don't want to, be negative, but it sounds like an engineering problem. Like, how do we have these constructs, and then how do we get them there to do what they want? But I just felt like, given that there were many ways that you could interpret the results, it wasn't obvious how to take those outcomes for refinement of the construct for the next round of animals, at least for us.
Well, I sometimes see it not only as an engineering problem, but also as a regulatory or policy problem. We're not using rat and mouse models because they're so fabulously good in predicting what happens in the human, but we're using them because that is what the regulator would like us to use, because that is the one we have to rely on and compare everything with. Terry, I wonder, while you have the microphone, and now you don't have it anymore, but you're still on. We had a question earlier, and that specifically was about your presentation, asking how many of those targeting, or how much of those targeting molecule would be considered as an optimum in targeted nanoparticles.
Yeah, so I think it depends on what you're actually delivering. So you need enough ligand on the surface that it's stable. Otherwise, there will be, if you don't have enough ligand, you'll have the particles will just crash out. There'll be some aggregation. Or sometimes you have to stabilize using polyethylene glycol, short chain polyethylene glycol, which is typically included in formulations for liposomes, lipid nanoparticles, things like this, just for circulation effects and for charge effects. And so we have found for some situations, if it's too densely packed, the ligands don't retain their function. There's just no way for the oligonucleotides to bind to their intended target. So we had to screen, and maybe this is where some of this machine learning would be better. Like, we had to screen high loading, medium loading, low loading, and it's a lot of work to make all those constructs and then to compare them side by side. But I would say it's just not obvious enough. Maybe Christine, you can comment on this, but I mean, you have famous people at your university who've been thinking a lot about the problem of reproducibility and how you monitor load and how you quantify things. Like, the data sets are just not good. Like, you can go through even worse than the polymer one. You go through the literature and it's just really difficult to know how we can help each other out, just related by the data that's being analyzed.
I'm with you 100%. It's there was a question also in the chat about what I think the most challenging part is in this AI and machine learning space for us right now, whether it's the models or using the models or the data. And for us, it's actually compiling these large, high quality data sets. And it's, as we all know, you go to the literature, even if it's in a fantastic journal, high impact journal, you try to reproduce the study, and oftentimes or sometimes you can't. And I often don't think there's any malintent. It's just the methods are not detailed enough, right? The information that's there is not detailed enough. So that's become super frustrating for us. In particular, when you think we got 40 papers out of more than 200. And we're just looking for basics like polymer molecular weight. Imagine publishing on a PLGA microparticle and not including the polymer molecular weight. It's so basic, but I think nowadays we're also inundated as reviewers. If you think of the last time you reviewed I don't know if anybody here is reviewing polymer papers, but if you are, I'm sure some of you are, did you actually look to see if the molecular weight was included? I mean, I don't know, right? You probably will now. If you are reviewing these papers, you will now, but I am now, I will tell you, but it is discouraging when we think of scientific literature and the challenge with kind of reproducibility or replication.
I would like to come back to that thought in a second, but I see that Samuel switched on his microphone, and I believe you have something to say on that topic as well, right?
Yes, so I think it's still linked to the talk that was given by Christine. So I think what I can say is that from my perspective, as much as I've got the institution that I belong to, which is B3, I'm also representing Africa, because I'm the current president of the African Materials Research Society. So we are looking at how we can promote excellence in all aspects of materials research. So one of the things that we're looking at is promoting nanotechnology in Africa, where we've got a program cooperation agreement with the United Nations Economic Commission for Africa. So what we've been doing over the past 2 years, we've been focusing mainly on 4 areas. There's nanotechnology, it's nanomedicine, nanotechnology for agriculture, water, and energy. So I'll just talk about some of the work that I know that's going on at one of the university in Africa, where there is this work on development of these sort of microemulsions that have got nanoparticles that use plant extracts as the reducing and capping agent that are typically used as topical formulations that are applied for skin cancer. So I know that the work that they do, you look at a range of plant extracts, and then you try that out on rabbits. So you find that in some cases you go all the way, and it's only when you get to the stage of testing that you realize that probably this does not work very well. So I'm just thinking that with the approach that Christine was talking about, that's another maybe sort of like an angle that can be taken to find a way of cutting down on the screening of the different plant extracts that you can have, because there's a certain way in which you can have an idea of the pharmaceutical phytochemical constituents for each plant, and then you can use that as a way of sort of screening in formulation. So that's a comment that I have. Thank you.
Many thanks, Samuel. I would like to go back to the to one of the questions we had earlier, also in the chat regarding standardization. Now, while standardization is obviously a good way to make things comparable all over, it is also a way of protecting inventions and innovations, and it can be used very much in order to exclude certain communities, and that also is something that we should not overlook in sustainability, especially as the sustainability goals are trying to bring people together, and behind those sustainability goals, the last goal is the one that is the community that implements all of them. So I wonder how you're feeling about standards and standardization. I want to look at DRAW, for example, because I know that for some of the very challenging stuff where you are working in interface of 2, if not 3 disciplines, isn't the problem of standards sometimes one where you don't necessarily know what sort of indicators you need? And so very unpopular quote, but still accurately, you have no handle of the unknown unknowns that you should be looking for in standardizing, right?
Yes, you're definitely correct. And standard is something very important, but I would like to share with you a story that we are doing during the last 4 months. It's in medicine segment, cosmetics, but it's showing us how we can share resources and building a standard for many things together. I'm leading in the EU a group that dealing with the correlative multimodal imaging, CMI. And we choose a project, something that it become necessary for medical doctors about tattoos removal. There are new techniques based on lasers, but it's not working due to the fact that you need to answer 3 questions. The type of the tattoo, the location, exact location of the tattoo, and the concentration. Otherwise, the laser cannot be focused and it's not making the work. We established, it's easy to use human being, but we would like, even during the pandemic situation, to measure the same samples all over Europe. We are using 23 different labs on the same samples. We prepared phantoms, mimicking tissue-like phantoms, 3 layers. We use artists, real tattoos, persons that inject different type of tattoos, inks, location, and we share it on all kind of tools, our own fluorescence as well as diffusion reflection goes to acoustic, optoacoustic, mass spectroscopy, all type of electron microscope, Raman, you can name it. And the idea is to build a standard how we can define the 3 main questions, type, location, and concentration, in order to solve it later on, maybe to build and aggregate different components. This is a correlative multimodal imaging idea, and to build a standard in order to remove the tattoos in the most efficient way. So from one direction, standards make things slower, but from the technical perspective, why not? If we can share resources, position, and people, we can solve things like I just described in this example. When we are linking different disciplines, that's it, the questions goes to my direction, there are totally different disciplines. Computer science or mathematics have no idea what are the nanomaterials, and the chemistry and the mathematics don't know what is going in the life science or medicine. So we need to be one of the adult responsive guys that know what is going on, but combining the correct technology will lead for new standards that will be good for all stakeholders in this story.
Super, thank you, thank you. We had another question in the written comments, and I think although you answered already, Christine, I think this is something that is well worth airing to the entire panel and sharing with the entire panel, and that is asking about the potential risks we're inviting by using machine learning and artificial intelligence to help us with the drug design. Now, my initial reaction would be bias, because you are still learning by largest data indicators. So I think the answer to that would probably be a tickling out of weak signals, but you are the expert.
Well, I'm still learning in this area as well, but I think you're right around bias in collection of or compilation of your data set. So where you're getting the data from, what you're considering good, reliable data and what you're not. And then limitations associated with the model. And of course, they have many limitations. As you saw, some, in some cases, we're able to predict drug release well, and in others we're not. And so then some of the model selection piece, or what we call a good model. And again, there's bias inherent in that as well. But, I think this is a field, there are a lot of people that are focused kind of around bias and ethics, around integration of AI and scientific research. So I'm certainly definitely not an expert, but recognize the need to kind of check my bias. And I think one of the criticisms actually, when I have presented on this, that one of the questions or criticisms that I usually get is, how can I trust any of the data from the literature? Am I choosing data that's only from groups that I know, or data from certain journals, and we're not doing that. We're very careful not to only choose data from groups that we know or data from certain journals. But I think there's obviously, as soon as you're starting to pick or hand pick or cherry pick or select, there's bias, right? Is this a good paper? Is this a good journal? One of the things that's been interesting is we are able to identify what could be outliers. So, we are able to identify where, let's say, PLGA and paclitaxel, there are so many papers on PLGA and paclitaxel, right? Microparticles, nanoparticles, and so on. And all of a sudden you get data that just doesn't make sense at all. The release curves that just don't make sense at all. And so then you can kind of draw a circle around that and say, okay, what's going on here? And so this could be used as an approach down the road to potentially identify data that are, where there's issues with the data.
Exactly, thank you. But I think you also made a very good point there, and I would like to come back to that in a moment, and that is about data collection and how much data do we need to store. And I'm going to use the bad word metadata here. But I'm looking at Terry here because, sorry, I'm always saying this, I'm looking at, you have no idea what I'm looking at because my camera is over there. So Terry, you actually mentioned one of the biases that is a bias without necessarily being able to know about it if we don't give the right additional data about the experiments that we did. You mentioned the enormous difference in circulation between female and male rats. So unless we actually ask researchers to complete a protocol that always mentions the gender and always mentions many other things, and I'm now going back to what we said before, we don't, in some cases, don't even know what sort of things we might want to know about once we find out that they were an influencing factor in a few years' time. How do we tackle that? I mean, surely you can't penalize your researchers for every 1 hour experiment having to sit down for 5 hours and log data. So what is it that we want? Is it a cleverer readout algorithm of all of our experiments?
Well, at least, I mean, for us, it was all very labor intensive. So we had no automation, that the reason that we included female rats is because there's been an emphasis, I think, worldwide, and especially at Northwestern, that the models that are being developed for medicine need to address the differences in the sexes, the biological differences, and usually it just hasn't been done that way. It's been just a single sex, and so I think acknowledge that. So the drug, if you're going to develop a generic formulation, then it has to be tested more widely, since there are actually distinct differences in how, just on a course level, biological males or biological females will respond to a drug. Yeah. I'm not sure how to solve that. I mean, that's the course level. That's sort of the easier problem, because the bigger problem that nobody talks about related to precision medicine is it's incredibly expensive. Because yes, you can develop something that will work for your particular set of DNA, but then everybody's DNA is different, which is why chemotherapeutics, only work about 60% of the population. And so that's also something that I think is important to think about. That the matrix gets bigger and bigger as we think about analyzing what could be important in the design of nanomedicine, standardization, and the model at which we're trying to test.
Exactly, but I sometimes wonder if we really need to have this what Christine coined earlier, reproducibility crisis, because we are talking about burning the equivalent of the heating infrastructure of small cities on creating bitcoins. Surely we're not going to have a problem to collect the data and to churn the data over if we really wanted to. And in this case, the innovation in medicine to save lives surely has to be a good way to do it here. So why do we have a reproducibility crisis rather than a data gathering and data management crisis? And the thing I'm hinting here at, Christine, is what I think you weren't able to show anymore was the automation that you have in processes. And through the automation, you can just tell a machine to log all of its metadata and all of its additional parameters that you might need in the future, to just log them until you decide that you need them. Why are we not doing that?
Well, I'm not sure. I guess I would say that my lab has just really started to integrate automation into some of the work that we're doing. And I think what really drove it, really drove us to this point, were cell culture studies. We were just doing ridiculous numbers of in vitro cell culture studies. And my students were actually getting sore thumbs and so on from all of the pipetting. And I say that, kind of laughing, because I remember all of the cell studies I did, and I didn't even have a multi-channel pipette at the time, years ago, obviously. So we've really integrated just very basic kind of using these Opentrons systems for automating our cell culture studies, and then some of our early formulation prep and analytical work. And it's been so helpful. And in particular, during COVID-19, like I said before, in terms of managing some of the physical distancing requirements, it's been great. But beyond that, it also reduces error, right? I mean, there's a lot of there's potential for human error. And what I love is, sometimes a student will do something and then we'll get the robot to reproduce it and see, if there's any difference in the data. Which is also one of the ways we've been able to validate some of the automated protocols. But right now, when it comes to really using some of these advanced, powerful AI machine learning models, we are limited by the lack of data that I would say that we have. That is our problem right now, is actually finding this high quality, large volume of data that we can use to feed into these models. And if you look at some of the drug formulation papers that are published out there, AI machine learning drug formulation, they're actually using relatively small data sets, which is concerning to me. I didn't even realize it was concerning until I got into this field. But we really need to be using data from thousands and thousands of formulations, not tens or hundreds of formulations. So we need high volumes of high quality data. And I think we'll start to adopt some of this, automation, digitalization. I think that's going to happen in the near term. And a lot of these systems are becoming very affordable now. So like I said, the Opentron system, and I don't have any stock in the company or anything, but I'm just saying super inexpensive and very user-friendly. So happy to share our protocols with folks if you're interested.
Perfect comment there. You gave me the keyword for my next question, but I'm looking around. Terry, switch on her microphone again.
Oh yeah, I just wanted to follow up on that, on your comment. Do you think, so, I mean, automation is doing wonders in catalysis, discovery, organic synthesis, and things like this. Do you think it's possible that automation and robotics can help build out your data sets? So you can start off with certain types of molecular weight polymers and whatever drug that you're embedding in it. And you can just do it, you're sort of like do it yourself, right? You know what the literature has produced, and they're incomplete in what they report. But if things can now be automated, where you can run hundreds of thousands of reactions, it seems like you can start to build your own data set that could test some ideas.
Absolutely. I think that's the direction that we need to go in. And I wouldn't have started there, because I would have said that we need to try to use the literature. We need to try to use what's already been published. I wouldn't want to, not be building on that kind of foundation. I mean, it's decades and decades of research, right, on things like polymer nanoparticles and polymer microparticles in particular. But absolutely, I think that's what we should be able to do. And then there'll be a data management issue, right? But, we're not there yet. We're just in the let's collect the data.
Thank you. And now I'm Samuel. Thank you.
Yeah, so I'm just wondering, probably just a question to Christine. So you're saying that the next phase is, in terms of the challenge, is the data management. If we say maybe 5 years from now, what would be your comment?
Pardon me, did you say about data management?
Yes, like predicting, looking at maybe 5 years from now, how much progress do you think we could make?
Oh God, I think we could make a lot of progress in a couple years. I don't think we even need 5 years. I think we start to adopt some of these strategies, and let's see where it goes. I mean, this is the latest and greatest. Let's see, I'm sure there are limitations, and some limitations we're not even aware of yet, right? As with every other kind of new technology or technological approach that comes along. But already I'm seeing the possibility of really cutting down on the formulation work that we're doing. We often have companies that come to us and they say, this is a drug that we want to formulate. And it's, you look at the drug just based on your own know-how, and you go, okay, should it be polymer? Should it be lipid? And so usually we can narrow it down to, let's say, polymer, and then we decide, or let's say lipid, whatever, but, and then we start to do the formulation work in the lab. And oftentimes I felt like it's a fishing expedition. So if we've got these data sets, and I think if we make them available online and other people can start to populate them, right? So your lab is adding data, my lab, whoever else's lab, as long as we have this minimum required information that's there as input features, and we're conducting our experiments well, whatever that means. I know that will mean different things in different labs. So that's going to be, part of the challenge always. But I think we're going to make a lot of progress in a short period of time.
What an upbeat comment, thank you. And I'm talking if nobody else is posting comments or questions, so please feel free to post more questions into the chat. While you are doing that, I'd like to come back to a point. There was one word that Christine mentioned earlier, and that was affordable. So affordability is also one thing that we need to think of under the sustainability goals, because it is meant to be inclusivity that we're driving. It is meant to be available for all. And one of the things I'd like to point out is at the beginning. Okay, sorry. The ability of female to do more than one thing at a time is highly overrated. I can clearly not talk and read at the same time. I think it was also Christine who in her introduction said that isn't it a great time to be in drug development when one is working with really cutting edge mRNA technology? And for all of the bad things that this pandemic brought with us, it has definitely brought a lot of chances to nanomedicine and nanomedicine's platform technology. And I think the word affordability is one that definitely fits for that one, because not only is it fast to adapt, it also allows us to work affordably in microreactors to solve the scale-up problem and to solve a number of other hurdle issues that the other technologies for vaccine development simply were not able to solve in that amount of time. Plus, it is more flexible in order to adapt to new variants of the disease. Nevertheless, I wonder if we have factored in the societal affordability enough, because it is exactly the fact that it is new and exactly what we as scientists are celebrating about nanomedicine and mRNA-based vaccines that has many people turning against it with the impression, the gut feeling, it is new and surely we don't know enough about it. But also the much deeper reaction to it of it has the word RNA in it, which sounds like DNA and therefore is probably related and must be bad. So I wonder, our quest of nanomedicine and sustainability, what are we going to do about the societal sustainability of some of our great innovations? I don't see any of my panelists switching on their microphones. Anybody wanting to step up?
I will try, and it's you are involving politics and it's recorded, so it's very dangerous what I'm going to say. The truth is that we are scientists. Scientists need to trust what they're doing based on their science, and we need to do all the protocols to make all tests, but mentioning some sequence of letters, maybe it's RNA, DNA, whatever, and worried about things that are new, it's definitely not science. And as some people worried about radiation from cellular technology and mixing with 5G technology and the issue of the pandemic situation, we need to be behind our beliefs, our finding. We know that this is true. We are eating food that it's much more dangerous than this kind of vaccine. We are driving cars that based on technologies that people can claim maybe it will not work, and just on the road your car will become parts and you cannot continue anymore. We are going to airplanes and we need to continue. And the main mission is that everyone that 60 kilometers around our university and our labs need to understand that what we are doing is for him, personally, for him. And if we will be a one big organ that everyone knows that it's true, this is what we can see in our eyes and to understand with our mind, it will be more easy to the big audience to understand what we have in our hand. But this is very shortly.
Thank you. That was almost good for a last word, but I don't necessarily want to take it away from the others. Let me just quickly check. Wojciech, you put your hand up. Am I right in thinking we are closing in 11 minutes?
No. I just want to make a comment, sorry, to your question. So with the social acceptance, it is a lot about the education. It is a lot about a modulation of the perceptions of the society. And this is actually on us as well. This is on scientists to go out and to speak the language of the society and actually educate the society and tell them, look, this is actually good. This is saving lives. And I think this is one of the missions of the network which we establish, the International Network for Sustainable Nanotechnology. It is to promote a science and it is to modulate the social perceptions to make sure that nanotechnology is sustainable and it will provide a sustainable future for us all. So this is what I want to mention in the context of your question.
Thank you. And you do confirm we still have a good 8 minutes before we close that and hand the word to you, yeah?
Yeah, indeed.
Good, super. That means that all of our panelists, and Samuel switched on his microphone again, get to talk about societal sustainability.
Yeah, so I think what I just want to bring out is this aspect of tapping into the indigenous knowledge. Was one of the things that is there is that once the society is aware of the benefits of nanotechnology, it makes it much easier for the communities to fully appreciate how nanotechnology can be used to sort of enhance what is already there. Because if I look at the context of what you can get from plant extracts, for example, I don't know how much you know about Africa. There is this part of Botswana where I am. There are these people that I remember Terry was talking about DNA and all. So these are people that that traditional hunter-gatherers. So they just live in the bush. Even when it's extremely cold, all they do, they're still putting on these sort of like just leather kind of clothing, traditional kind of clothing that they put on. And I know that they've got all sorts of extra that they get from plants. If someone is stung by a termite, they can just take off it. Even when you talk about when the women are giving birth, they've got a way in which you just chew some roots and then delivery is just smooth. So there's a way in which through nanotechnology, you can tap into some of that indigenous knowledge and get the most out of it and make a difference going forward. So I think that is something that if it's put across in such a way, so that it's sort of enhancing what is already there, definitely I think it can be received positively. Thank you.
Thank you. And that is also a great example of inclusivity. So you are not saying that you have to eliminate all of the old traditions and teachings. You can actually learn from them, because quite often it turns out that the plants that our ancestors have been using actually have a functional component that can then be extracted and or synthesized. So I think that is a great way to bring it close to people, that we are not eliminating the old for the new, but that the old is building on the new, and thereby marrying the world of tradition and innovation. Christine and Terry, I wonder if you have a comment on affordability and sustainability of society. Sorry, societal affordability and sustainability.
Yeah, so I'll just comment briefly on what he said earlier. Is nanotechnology, and as its dedicated field, if you will, as it's matured over 20 years, there is a huge investment in integrating with the public, because we, at least in the United States, we wanted to avoid the debacle that happened with genetically modified foods. So there wasn't enough public education and engagement, and so Nano, did its own thing. But what has been sort of stunning to me is it's not just the science. You know, if you're talking about societal impact, I mean, you can have all sorts of science like we've seen during the pandemic, and it's interpreted very, very differently by whoever wants to interpret it. So I think there are some key challenges to educating the public, and I don't have a good solution for that. I'm in all sorts of areas related to all the different hats that I wear and people that I interact with, but I think that's a non-trivial matter. But in terms of affordability question, I actually think the affordability question is related to innovation. So I have been consulting with some organizations that want to bring, you know, a product to market, but they need to do the analysis in such a way that it can be distributed over many different labs so they can test for reproducibility. But if you require multi-million-dollar pieces of equipment to do the analysis, that's never going to work in terms of, I mean, the company will not ever be launched. You know, the product will never go forward. So the idea of what is the minimum standard for characterization and tools, that makes it more affordable to those of us that have limited resources, but also it's commercially important because related to innovation, because if the innovation cannot get out, you know, because it can't be validated, then that's its own set of problems. So I think actually thinking about affordability will increase ideas and innovation.
Thank you. Christine.
So many great comments that I'm thinking about. First of all, I just think it's fantastic that we have so many scientists and representatives from different countries today, and all of the different ideas that have come forward. And we think about the 17 SDGs and how each of them are important. And you start to address one, and perhaps then you're actually, you know, making things worse or moving backwards when it comes to another. And so important to see all the different comments that have come up in the chat and that have been vocalized here today. I think during COVID, when it comes to education, that is absolutely critical. And as scientists, we often forget that we have a responsibility to educate the public. And we've seen the power of misinformation, disinformation throughout COVID. We've been dealing with it here, frankly, in the last few weeks in my province in Ontario with a number of different protests and things that are going around. Very, very challenging time, continues to be a challenging time. And more than anything, we see the power of communication, of accurate, clear, accessible communication around science. Beyond that, when it comes to affordability, one of the things I think as scientists, many of us don't have access to the knowledge that we need to really start to think about whether or not what we're working on is actually something that could be implementable. You know, when it, you know, economically, clinically, is this something that, you know, is likely to be adopted by clinicians, likely to be accepted by patients? Is it affordable? One of the things, I run a National Center of Excellence on nanomedicines, and we've recently set up this economic health therapeutics assessment platforms. We have scientists with expertise in that space that are working with our biomedical sciences now to help us understand, you know, whether or not what we're actually working on, you know, has market potential. What's the competitive landscape? You know, what would it cost to actually manufacture something like this? How many ingredients do you have in your formulation? And I think as scientists, sometimes we can get really excited about making things complicated as a means to achieve novelty. You know, it's novel because I've got a nanoparticle that instead of having 10 things dangling off of it has 12 things dangling off of it. So it's new. And it's incremental really in terms of progress and is likely to go nowhere when it comes to making a clinical impact from my perspective anyways. And so I think simple is always best, but that may not be exciting and may not get you into the highest impact journals. But when it comes to drug formulation and clinical translation, that's best, right? 3 formulation ingredients is better than 280. So there's a lot of things that I think are kind of working at odds with one another, in particular, kind of in the nanotechnology drug formulation space, which is the space that I come from, that we have to think about. And, you know, when it comes to affordability, and somebody made the comment, I think it was our moderator, who's been absolutely fantastic around ensuring that if we implement minimum standards, or if we go with standardization in a field, that we ensure we don't exclude certain, people in certain regions. And this, of course, is also extremely important. And so, has to be considered, and we would have to, if we were to implement standards or minimum standards that would be reported on a nanotechnology or a microtechnology or whatever it is, we would have to have people from different regions that would, approve the standards or contribute to the standards. Anyways, I just wanted to say, because I know we're running out of time, how fantastic this session has been, and just so powerful to hear these thoughts from people in different regions. And in particular, Samuel, really delighted to hear about the research in your region. So thank you for sharing that with us today.
Thank you so much. And if you allow me, I'd like to just have 2 minutes in order to do a quick summary of what we just heard in the last couple of hours. And then I give the floor to Wojciech to close this session. We started off looking at the stars, and I think we've sort of done full circle and are doing that again. We looked at nanostars, and we looked at nanostars specifically with regard to what great many parameters they have to tweak and tinker with when it comes to designing them to be safe and to be more sustainable, but to still do what they are supposed to be doing, and that is their functionality in nanomedicine. And we moved from there to actually pick up on that by looking at what computers and digital methodology can do for us to use all of these different parameters, because just the shape of a star and the loading with its ligands already poses a formidable number of parameters that need a computer in order to work through all of the different iterations of it. And we heard Christine then talking about the way that computers and automation can be used in essentially making predictions easier for us to handle, but obviously throwing up a number of different challenges as well. And some of these challenges that we know we have at the moment is this reproducibility crisis, which I still believe we should be able to compensate by just collecting all the data that we possibly have, but then we may be creating a data management and a data storage crisis, at least temporarily. But in essence, the biggest challenge is being predictive enough in order to also care for areas like the ones that Drew is working in, and that is the interface of large areas of unknowns that happen, and therefore not knowing now what could be an important data point tomorrow and what would be some a correlation that we might like to go back into and look at it because of something that we find out, some sort of phenomenon and context that we find out only in a few years' time as the knowledge increases. And all of that needs to be seen in front of the aspect of making all of this green and sustainable. And it was Samuel who brought the great example of wedding the current technology and quest for innovation to the old traditions and learning from them and making sure that we are being inclusive in that, and we are not doing what Christine coined so nicely at the moment by just throwing more complexity at something and trying to prove that surely is something more innovative because it is more complex. And that brings me to one of my pet topics, and that is that the regulators also have some responsibility here. Christine just assigned responsibilities to us as scientists. We need to be more open, more inclusive, and we need to engage with the public and with every individual more. I think Drew was quoting the radius of 50 kilometers. I think if every scientist can reach the relevant people in a 50-kilometer radius, we would not have all of these science skeptics these days. But regulators also have a responsibility because they tend to react to this spiral of innovators make something more complex and believe that makes it more innovative. they follow suit with their requirements for safety evaluation. And I think you probably see the same for your clinical trials as well. If you make a molecule more complex, then you have to go through another round of evaluation just to show that it now doesn't do something that it didn't do before. And that is something that we need to stop as well, because there's also a question of unmet affordability in the regulatory regime, which just makes our innovation incredibly expensive these days. But in all of that, and for all of these different challenges that we've thrown up, I'd like to go back to the last statement, an earlier one that Dror made. And I think he, in essence, said that we should not give up. And I particularly look at the young scientists that we have around the table now. We should not give up then all of this struggle that we have at the moment. And every time we think we've cut one corner, out comes another big challenge. All of this struggle is a sign of the pot of gold that comes at the end of a formidable challenge. And we are doing this in order to improve ourselves. I think I've sort of paraphrased you correctly, Dror. And in that, and I've spoken longer than I thought, I give over to Wojciech.
Thank you so much, Steffi, and thank you so much, everyone, for participation. Great presentations. From my side, just 2 sentences for each of the presentations. Terry, corona, protein corona, you said Achilles heel. I think it's also the holy grail. If we understand how the corona is transforming, moving, when the particles are moving between, between the organs, we can actually develop something which is precisely targeted. Christine, I'm from the Sydney School of Pharmacy. Thank you for saying that there is no better time than now to be a formulation scientist. It is a music for our ears, myself and few colleagues on the call here from our school. Thank you. And thank you everyone for participation. Thank you panelists. Thank you for your time dedicating to these conversations. And we are certainly inviting you for the summit, which will happen in July. Each of you will receive the invitation from us. Thank you again for very productive time, last 2 hours. Thank you and enjoy the rest of the day.
Thank you.
Okay.